A virus can take one of two pathways when it infects a host cell. And what I'm going to do in this video is discuss the lytic cycle. And the first step in the lytic cycle is the attachment of the virus or the phage onto a host. And once the virus attaches onto the host's membrane, it's going to inject some genetic cargo. Viruses are incredibly diverse. This genetic cargo can either be in the form of single or double-stranded DNA or RNA. But what's going to happen is that genetic cargo will be now in the cytosol of the host cell, or the cytoplasm, and what it is going to do is cause the production of something referred to as hydrolase. And hydrolase is an enzyme that will degrade the host genome. And so the thing you have to remember with viruses is that they are incapable of reproducing themselves on their own. They need to hijack another cell's machinery in order to replicate themselves and their genetic material. And so in the case of viruses, how they're going to be replicating their genetic material is by using their host cell's own nucleotides and by breaking these nucleotides apart, the adenine, thymine, guanine, cytosine, uracil, and it's going to rearrange the nucleotides such that they can be used in the virus's progeny. So we see in the very first step of the lytic cycle how important it is that the virus have a host that it can use, especially for its own genetic material. And so after the hydrolase has done its job, what we now find is that the host genome is degraded, and once the genome has been totally broken down, we actually make copies of the viral genes. And once the virus has made copies of its own genetic material for its progeny, the next step is going to be the production of the outside features of the virus, the sheath and the base plate. These are going to be the things that actually attach onto future host cells to keep producing more viruses. And we're also going to be forming the capsids, and the capsids are what's going to store these copies of the genomes. And after it goes through this process, I'm going to replicate the chromosomes, and now we're going to begin assembling the new progeny of the virus. And once the progeny have been created, the final step in the lytic cycle is going to be the production of something called lysozyme. And so lysozyme is an enzyme that will weaken the cell wall of prokaryotes to such an extent that it becomes permeable. And it becomes so weak that the viral progeny inside are going to be released into the environment where they can actually infect more host cells and continue the process. And so from a high level, this is what's going on during the lytic cycle. Now, the key thing to make note of when you're learning about this stuff is the fact that the host cell dies. And if you're a virus, while you did just make a bunch of copies of yourself and you've exported those copies into the environment, the problem is you can't make any more from that host cell because you've just killed it. And so what we're going to find later on is that there's another pathway that a virus can take called the Lysogenic Cycle, in which it will embed itself into the host genome and remain dormant such that it can produce yet more progeny. And so it's pretty cool. I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.